Fantastic. Okay. So good evening, everyone. It's a real pleasure to be here with you today. So I guess really the purpose of my presentation is to give you an overview of the organoid technology. So define what is an organoid, and given the increase in the use of non-animal models in research and in translational research as well, what the Monash scientific community decided to do in response to this, and therefore the development of the Monash Biomedicine Organoid program in 2018. And finally, what we've done to basically use the organoid technology to support translational medicine for patients with breast cancer. So I'm going to cover this briefly, 3 points tonight. So to start with, I would like to share with you this very basic figure that is really summarizing the number of scientific publications that have been released every year for the last 23 years. And I've basically used the online database called PubMed to search for these scientific publications, and we include the word organoids in the abstracts or in their work. And what is quite striking here, and then you can really see that for the first 10 to 12 years, or just a quick question, can you see the mouse when I move it on the years here? Or do you see something or nothing?
I can see your mouse, yes. Yep.
Fantastic.
Yep.
Amazing.
Thank you.
For the first 12 years, you can see that the organoids in general were not necessarily used by scientists. And therefore, you can see that pretty much there was no scientific publication that were using this model in the world. But suddenly, around 2012, 13, you can see that there's a dramatic and a striking increase in the number of scientific publications that were using this technology, which was really important and is really showing to us that this model and the organoid technology is really the technology of the future. And I didn't put the 2024 data, but it's even more striking. We are getting to nearly 5,000 publications that are published every year now, and it's going to keep increasing. So this technology is so essential, I think, for fundamental research, but also for translational research, that in 2022, there was a very important event that you're probably aware of, is that the President of the United States, Joe Biden, signed and approved the US Food and Drug Administration Modernization Act 2.0, which refutes the Federal Food, Drug, and Cosmetic Act of 1938, which mandated animal testing for every new drug development protocol. So before 2022, the traditional pipeline for drug discovery and providing these drugs for clinical trials in patients was to validate and perform preclinical tests using human cell lines in labs that are grown in plastics. So you would do your validation in labs, and then eventually you would have to do a mandatory animal testing. Once the drugs have been, the efficacy, the safety have been tested in animal models, it would be then basically given to humans during clinical trials. The main issue with this pipeline is that 90% of the drugs that were given to patients failed. And they failed because of safety issues, inefficacy, toxicity, side effects, really suggesting that using animal models to validate drugs that were supposed to be used in humans for humans was not an appropriate way of doing research. And therefore, they've done this, they've approved the use of non-animal models and human models to validate drugs and bypass the use of animal models and provide these drugs for patients. And some of the non-animal models are listed here on this figure. So you can see that you've got organ on a chip, tissue engineering models, AI approaches, and in silico models, and finally the organoid models. These drugs, this meal now is providing scientists, a way to use these models to bypass these animal models. It's extremely powerful, and this is quite new. It's been approved in December 28th of December 2022. There's a second point that I would like to make here, which is related to the impact that these non-animal models and the strategy for developing products in Australia could be very essential for our economy, because we are actually strategically placed in the world to be at the forefront of the organoid technology and non-animal models technology. And indeed, if you look at the CSIRO report that was released in 2023, it is estimated that there will be $1.3 billion in revenue for Australia by 2040. So really suggesting that both in terms of ethical considerations to improve, clinical translational research, but also providing new source of revenue for Australia, it's very essential to develop the organoid technology. So if we go back to the first figure that I put on the first slide, what I believe was really a key event that led to this massive use of the organoid technology in the world was actually this paper. And for an intestinal gut biologist, it was a mind-blowing discovery. What Hans Clevers and Toshi Sato, who are the leading authors in this team, discovered is a way to reproduce in labs the function of organs. And for these specific papers, they were able to reproduce the function of the gut. So briefly, we need to cover, the way the tissue is organized and the gut is organized in healthy people. So I put on the left-hand side here what would be a section of the human small intestine. And what you can see here is I've colored with multiple different colors these cells here, and these cells are actually facing the lumen of the tube. So if you consider the gut as just a simple tube, what you can see here is that you've got millions and billions of cells that are just facing this tube. And these cells are organized into two really distinct compartments that you can see here. These small compartments that we call the crypts, and this very large compartment they look like finger-like projections, and we call this the villus. And they have two distinct functions. The villus are really associated with absorption of nutrients from food intake. So once the food is digested, these specialized cells in the villus would absorb nutrients. In contrast, the cells that are located at the bottom of the crypts in these little pockets are really essential in cell division, really making sure that they provide and sustain and they create new cells. And this is done because at the bottom here, I've labeled in green these specialized cells that we call the stem cells. And in all our bodies, in all our guts, in humans and animals, we have stem cells. And as we speak today, and by the end of my presentation, these stem cells would have generated billions of cells in our guts in just 1 hour. So it's one of the most active tissue in the body. So for decades, scientists have tried to replicate this architecture in the labs and generating, I would say, in vitro models. But it was extremely challenging to really replicate these features until Toshi Sato identified culture conditions that were essential to sustain the self-organization of these stem cells into mini guts. And what you can see here is actually an organoid. And this organoid is really simply a mini avatar of the tissue of interest. So in this case, you can see that they form these little domains that contain all the cells that you would find in the tissue. So you do have these green stem cells specifically located in these crypt domains, but you also have these cells that are involved in absorption. So in a way, when you think about this model, they really recapitulate the feature of the tissue, both because they contain different cell populations, but also because they also maintain the function of the tissue. So in simple words, they are just mini tissues that we grow in the lab that form 3D structures. And just to demonstrate that, what we've done here at Monash was actually generating these organoids, but we've also labeled these organoids with different colors. And what you can see here is a 3D reconstruction of an organoid. And we've labeled in green here the cells that have the ability to grow and to form new cells. And you can see that all these green cells are restricted to these CRIB domains. And this is exactly where you would find these cells, and these cells have the ability to grow in the tissue. They will be restricted to these CRIB domains as well. I've also labeled these cells in red, so other cells in red. So all these cells that you can see here that contain this red label are actually stem cells, and they are indeed also located just at the bottom of these CRIB domains where they would be found in the small intestine. So it's a very, very nice model. Just to show you again another movie that really suggests that these organoids mimic the original organ, we've been able to label specifically in green here the stem cell population. And we can see that they are really located in these little domains, and they are restricted to this domain. And the way they grow is quite, in this context, very fascinating. But you can see here, if you look at the left-hand side in this picture, that these stem cells are indeed really located just at the bottom of these little units that I mentioned in the small intestine in human. And we can recapitulate that very nicely in this model. So this is just an example of an organoid that is formed from the human small intestine. And over the years, scientists around the world have been able to derive these organoids from multiple organs. And I've just listed here the main organs, and the main organoids have been generated. And we contributed to the development of the mammary gland organoids and breast organoids. People have established liver organoids and brain organoids, as well as lung, stomach, and intestinal organoids. And we also have a strong expertise in intestinal organoids and stomach organoids. So as you can see here, I've listed at the bottom of my presentation, both on the left and the right-hand side, all the publications that refer to these organoids. And you can see that it started in 2009 with Toshi Sato's study. And then pretty much every year and every couple of years, you've got a publication of new discoveries related to these different organs. And the field keeps expanding, and there's more and more organoids that are generating for pretty much any organs and any tissues in the body. So I guess in response to the expansion of this organoid technology in the world, we decided at Monash to develop in 2018 the Monash Biomedicine Discovery Institute's organoid program. And really the point of the program was initially quite simple: to provide and to make sure that the technology was available to scientists in Australia and to support these scientists throughout the development of this technology. So we've done that by providing optimized reagents to the scientific community, providing facilities and equipment and full laboratory assays to colleagues and fellows to make sure that the technology was employed throughout Australia. We've made some technological advancements as well. And I think most importantly, we've also developed in collaborations with clinicians, medical oncologists, several key bioresources. And I will discuss that at the end of my presentation. One of these key bioresources is breast cancer organoid bioresource, and the way we're using this bioresource to tackle precision and develop precision oncology in Australia. And we're also involved in providing consulting advice, training for PhD students, more advanced researchers throughout Australia. So our team at Monash is composed of my manager, Dr. Eglanty Ballon, the senior research officer, Dr. Sarah Randall Denlow, two research officers that are really associated with the breast cancer organoid project, Dr. Dilys Lung and Dr. Jasmine Akor, and the research officer is really here to support, to generate some of the key reagents that I mentioned before, and provide advice and assistance to PhD students, Ms. Joanne Hu. And I guess over the years, if we go back to this, the list of organoids have been generated worldwide. We've contributed to these efforts by developing both organoids from normal breast and breast cancer. We've done similar work in the bowel by generating normal colon organoids and cancer, colorectal cancer organoids. We've also generated organoids from babies with an inflammation in the gut called necrotizing enterocolitis, or from adults with Crohn's disease. And we've also been able to generate organoids from animal tissues, such as mouse mammary gland organoids, small intestinal organoids, colon organoids, and stomach organoids as well. And to show you some examples of these organoids, I'll put some pictures on each side. But I think it's really clear when you look at videos. So these organoids, it's an example of mouse small intestinal organoids, and it's a recording of the growth of these organoids over 4 days. And what you can see, initially they form these very small pockets, but eventually, very rapidly, these pockets would collapse and start forming this budding domain that I mentioned before. And these budding domain are essential because they contain the stem cell population that I described before. What is very, I guess, outstanding with this model is that with just one culture like that, so you can basically expand the culture and keep that in your labs pretty much forever. So just to give you an example, when I got my PhD in France from the Faculty of Medicine, and then I did a postdoc position in the School of Bioscience at this Cardiff University. And I was generating intestinal organoids from mouse tissues. And I managed to basically keep these cells in culture for nearly a year. And the system was quite simple. Once they reach this size here, what you do is once they reach this maximum size, you break them into small pieces mechanically, and you put them into new plates. And within 5 to 7 days, they would form new organoids. And you can keep basically breaking them up into small pieces and re-putting them into new plates to generate and expand these cultures. And you can do that forever, in theory. So we can do that with, it's an example for, mouse tissues. We've done the same with human colon organoids. And again, they are forming these spheres. If you break these spheres into small fragments and you put them into new plates, it would generate new organoids, and you can keep them in culture pretty much forever. The beauty of this system is you don't necessarily need to keep them in your lab. You can also put them, cryopreserve them. So put them in your fridge, not in the fridge, but in your freezer at -80 degrees. Leave them here for several years, and you can defrost them, and they would form new organoids. So you've got access to a source of cells from different organs and different species, and it's an unlimited source of cells. And we've done exactly the same exercise here, but using human colorectal cancer tissues. And you can see an example of these organoids that are growing in the lab. It's on the right-hand side. And we've been actually able to generate one of the largest bioresource of human colorectal cancer organoids in Australia. And the last thing that we're doing, and I think it's also very essential, function of the organoid program is, consulting advice, but also training, and training, the future generation of scientists, including, PhD students and early career researchers. And we generally do, I would say, 1 to 2 sessions a month to train basically scientists all over Australia. And And I would like to mention it as well that we are organizing every year a research, a method-focused organoid symposium here on site at Monash Clayton. And I would like to thank you for your support, because in 2024, we were able, thanks to your support, to provide actually prizes to the best poster presentation from PhD students and the best poster presentation from early career researchers that presented their work on organoid research, and were basically awarded these AFSA prizes for organoid research last year. So to basically move on, because I don't think, I mean, it's interesting to discuss about the organoid program, but I think you have an understanding now what we are doing. I really want to change gears here and really now explain the way we can use, and we've used the organoids to support precision oncology and personalized medicine. And I will start by, really describing the breast cancer, the disease, mentioning that it's an extremely common disease in Australia, and we know that 1 out of 8 women will be diagnosed with breast cancer in their lifetime. And unfortunately, it is estimated that there will be around 3,000, more than 3,000 patients that will die of breast cancer. So it's an epidemic, and it's a very dramatic disease impacting, thousands of patients. So currently, patients are pretty much, there's a one-size-fits-all approach, where most of the time patients are treated with breast surgery. And in the case of advanced breast cancer, it will be a combination of radiation therapy and chemotherapy. And the most common type of chemotherapy that will be given to breast cancer patients would be a combination of 3 chemotherapeutic drugs that are called doxorubicin, cyclophosphamide, and paclitaxel. And this is the case for advanced disease. I'm only talking here about advanced disease. Advanced disease means that the cancer cells have started migrating away from the breast. For example, they've started moving from the tumor that was located in the breast to the lymph nodes, or to other organs such as the liver, the lungs, the brain, or the bones. And when we give chemotherapy to patients with advanced disease, we don't really have a way to predict whether the chemotherapy is going to be effective in treating patients. And if we take an example of this cohort of patients here, we have 10 breast cancer patients. And what we know currently, if we give this standard treatment, so this standard chemotherapy, doxorubicin, cyclophosphamide, and paclitaxel, we know that the cancer cells for these patients are going to respond to the drug in 50% of the cases, which means that in 50% of the patients, there will be a clinical benefit. The cancer cells will be killed, and therefore the tumors are going to decrease, and potentially patients, in some cases, can be cured. However, what we know is that for the other 50% of the patients, there's actually no clinical benefit, because the cancer cells are not sensitive to the drugs, but the patients are still undergoing these severe side effects associated with chemotherapy. So it's actually extremely difficult for patients that are going through chemotherapy if there's no benefits, if there's no clinical benefits. So what we are aiming to do is develop a model where we can give the right drugs to the right patients. And we call that precision oncology, or precision treatment, where we want to find a way to identify which cancer cells are going to respond to chemotherapy, and whether this chemotherapy is going to be beneficial for one group of patients, and which chemotherapy would be, or which type of chemotherapy would be beneficial for another group of patients. So really going into the concept of getting the right drugs for the right patients and for the right type of cancer cells. So the concept of precision oncology. And given what I've introduced before, you can start understanding where I'm going now. And we believe in the scientific community that we could use the organoids to answer these questions. So what we've done and what has been quite nicely shown in the scientific community is that if you take breast cancer tumors, so tumors from breast cancer, that you dissociate, that you cut into very small fragments, and you put these fragments and these cells in a matrix. So the matrix is pretty much some sort of a 3D jelly solution that will allow cells to adopt a 3D conformation. So instead of forming layers like it was done traditionally before, when cells would grow on plastic and just form layers of cells, in this case they can form three-dimensional structures, really recapitulating also the structure of the tumor. So you put them into a matrix, and then you feed them with specific cocktail of growth factors. And if you wait for 2 weeks, 2 to 3 weeks, these cells will turn into mini tumors and avatars of the tumors that we call patient-derived breast cancer organoids. And what we've been able to do here at Monash is generating a biobank of these organoids. And this is a collaboration that involves many scientists across the Biomedicine Discovery Institute, but also medical oncologists, surgeons, pathologists from Cabrini Health and Tissue Path. And I think I just wanted to put here the pictures of the main drivers of this project. Gary Richardson from Cabrini Health, with a medical oncologist, Dr. Jasmine Kaur and Dr. Delis Lang, have been essential in basically liaising with surgeons and scientists at the BDI to develop this technology. And of course, we've been involved in developing that. So to briefly walk you through the actual protocol, the way we generate these organoids, what happens is when a patient is diagnosed with breast cancer at Cabrini Brighton, Dr. Peter Gregory would perform removal of the tumor. And of course, we have ethical approval to do this work. And the patient would also sign consent to provide these tissues to generate organoids. So it's ethically has been completely discussed and covered. Dr. Peter Gregory would remove by surgery the tumor, and the pathologist, Dr. Tim Nuttall at Tissue Path, would take slices of the tumor for his pathology report, and then whatever is not used for the pathology report, we would get this tumor sample that we bring to the lab. So we go there, we collect the tissue, and we start by dissecting the tissue into very small fragments. So very small fragments of the tumor that we keep digesting with different enzymes. The point is really to go from the tissue to very small cellular fragments that we isolate and that we seed in the 3D matrix, the 3D jelly that I mentioned before. And then once we apply the medium, we can generate breast cancer organoids for individual patients. So these are 15 pictures of 15 different breast cancer organoid lines that have been generated from 15 patients. So pretty much, we are in a position now to establish organoids for each individual breast cancer patient that will come at Cabrini Brighton. And over the years, we've generated more than 100 breast cancer organoid lines. And if you want to look at this movie, it's really showing you the way they grow. So each of these individual structures, these balls, are actually breast cancer organoids, and we've imaged them over, I think in this movie, 7 days. So you start with these very small structures, and they keep basically growing to the point where, sorry, you need to break them into small fragments, put them into a fresh matrix, and keep expanding. And we've been so this is an example of a breast cancer organoid lines established from patient 14, so called HBC 14 for human breast cancer organoid 14. And we've been able to keep this line in culture for nearly 6 months. And at some stage, because we didn't need to amplify further, we stopped them in and we put them in the freezer. But we could have kept these cells in culture for way longer if we wanted to. So what is very essential here for us was to be able to generate organoids from multiple types of breast cancer. And I'm not going to go into the details here, but I just want to mention that in general, breast cancer is divided into different subtypes. And these different subtypes are associated with different clinical outcomes. But what we know is that these subtypes are categorized into the luminal A or luminal B subtypes, the HER2 subtypes, or the triple negative breast cancer. And what you can see here in this figure is that we've been able to establish organoid lines from all these different subtypes of breast cancer. So we have access to a bioresource that very nicely recapitulates breast cancer heterogeneity. So all the different breast cancers that exist in patients. And over the years, we've been, our success rate is currently around 70%. And I think initially it was quite challenging for us. We had to change protocols. We had to make some changes in the medium that we were using, the matrices that we were using. But now we are more around the 80% success rate. So which means that we are able to generate, in most cases, breast cancer organoids. And this is aligned with this publication. This is the original publication from the group of landscapers that described this protocol in 2018 and suggesting that in this paper they can achieve organoid establishment from tumors at a 60% success rate. So now we are higher actually than this reference paper. So one important point here is that you can ask the question, okay, you can generate organoids from these breast cancer tumors, but how do you know that these little organoids are actually faithfully recapitulating the tumor? And it would be a very fair question. And in order to answer this question, we've used a very complex approach, and I'm not going to go into the detail of all the different techniques that we've used, but we've used what we call the multi-omics approach to characterize these breast cancer organoids and compare these breast cancer organoids with the parental cancer. All these methods, the point of all these methods was really to compare the tumor and the matched organoids. And to do this, I'm just going to give you one example, is looking at the mutational profiles, where we can characterize and analyze the mutations that are found in the patient tumor and look at the mutations that are found in the organoids that are derived from this tumor and see whether they matched. And we use a technique called whole exome sequencing, where we can analyze all the mutations that are found in these cancer cells, all these aberrant mutations, and compare them. So in a very nutshell, we've done that across 16 tumors and 16 organoids, matched organoid lines. And this figure is actually very simple. What you can see on this axis here is the driving mutation. So the main mutations that are found in the tumor. And on the x-axis, so at the bottom, you can see the patient code. So going from patient number 2 to patient 41. And I've got 3 different colors in my presentation in this schematic here, showing basically the mutations that are found in the tumor and in the organoid, so both in the tumor and the organoid in blue. The mutations that are found solely in the breast tumors, or the mutations that are found solely in the organoids. And when you look at this figure, what you can see that most of the time, the mutations are conserved in the organoids and are shared between the primary tumor, between the breast cancer, and their matched organoids. Really suggesting here that when you establish these mini tumors in the lab, they faithfully recapitulate the genetic features and the mutation profiles of the tumor they come from. And I don't really want to cover all the other points, but it was just to demonstrate that indeed at different levels. So I've discussed about the DNA mutation profile. We've also looked at other features, and pretty much the organoid is a very good model of their parental breast cancer. And the last point I would like to discuss here is to go back to my, this part where I discussed the concept of precision oncology. Can we use then these organoids to perform a drug screening and identify the best drug for the four unique breast cancer patients. And therefore, I'm just going to finish by mentioning the drug testing approach and what we've been able to do in the Organoid program. So I've mentioned to you originally that patients with advanced disease, with an advanced breast cancer, would receive a combination of chemotherapy, and in general, they would get treated with a combination of doxorubicin, cyclophosphamide, and paclitaxel. And what we've done here is establishing organoids from patient 5, patient 30, and patient 35. And in real life, these patients were also diagnosed with advanced breast cancer. And these patients got treated with doxorubicin, cyclophosphamide, and paclitaxel. So what we did in the lab was generating these organoids, these cancer organoids, and treating them with the drug doxorubicin. And what this figure here is showing you, in yellow here, you can see the concentration of doxorubicin going from no doxorubicin on the organoids to very high amount of doxorubicin, suggesting that we are exposing the organoids to this very large concentration of doxorubicin. And what you can see here, and as expected, when you've got the reference here, it's 1, it's showing us that they are healthy organoids, healthy breast cancer organoids. But the more doxorubicin you put in the system, you're more likely to basically kill these organoids, to the point where, with 10 micromolar of doxorubicin, most of the organoids have been killed. And if you look at the picture here below, you can see that these untreated organoids look very healthy. You can see these little balls of cells here in 3 dimensions. But when you look at the doxorubicin, when we apply high concentration of doxorubicin, so 10 macromolar, there's no organoids forming, and you can only see cell debris and dead cells. But what is quite striking in this schematic is actually that the 3 different organoid lines respond differently to the drug. You've got the blue organoids here that seem to respond very well to doxorubicin, and therefore, if you look at this concentration here, they are more sensitive to the drug than the other line. And in contrast, when you look at the line, the organoid line number in green here, HBC 35, with this concentration of drug, 1.01 micromolar, you don't really kill them as much as the other line. So you can really see a difference between line 35 and line 5, being 35 seems to be more resistant to the drug, so less sensitive to the impact of doxorubicin versus HBC5 that seems to be more sensitive to the drug. And what we are aiming to do with this tool is basically link this data that we've obtained in the lab with the patient data. So we are in the process of pretty much waiting for the clinical outcomes, whether the patient responded to the drug or not, and then we'll basically match these data with our data that we're generating in the lab. So it's a work in progress. I don't have the final outcomes yet, but I'm really hoping that we'll be able to share that with you in the future. I wanted also to really describe you a study that was published by a team in China led by Xuanjia Deng, and that was published in Advanced Science in 2021. And they pretty much applied a similar strategy here. I decided to use organoids from breast cancer patients to identify the best drugs for this specific patient. So we're just going to go through a case study based on 1 patient, but there are more patients that were treated using this strategy that are listed in this paper. So here you can see patient 148 diagnosed with a triple negative breast cancer with tumors that were found in the breast, but also in the lymph nodes. So these patients were treated with chemotherapy first on the 16th of April 2016. However, what you can see here in red is that the chemotherapy was not efficient because the disease progressed. So we call that a progressive disease. So chemotherapy was not efficient, but they still performed surgical resection to remove the tumor and also the lymph nodes here, where that cells invaded the lymph nodes. They performed radiotherapy in July 2016, and they were monitoring these patients. Unfortunately for this patient, in January 2017, multiple lung metastases were found, suggesting that the treatment was not sufficient to prevent the formation of new metastases in the body. So what they did here is provide another type of treatment called capecitabine, which is again chemotherapy-based treatment. However, the disease was still progressing and the chemotherapy was not efficient. So what the clinicians in collaboration with the scientists did was to collect these breast cancer cells that were found in the lungs, and they grew organoids out of it. And they performed a large drug screening with all the drugs that are currently approved by the FDA, so the Federal Drug Administration, Food and Drug Administration in the US. And by doing that, they were able to identify a combination of two drugs called everolimus and vinorelbine that were able to block the growth of the organoids in culture. And what they did was then to provide this drug to the patient. And in contrast to all the attempts that were tested before that didn't work, the combination of everolimus and vinorelbine were able to at least stop the disease. And it was called a stable disease, where the disease doesn't evolve further and the tumor cells do not form bigger tumors. So that's what we call stable disease. So by using this approach, scientists and clinicians were able to, at least for this specific patient, prevent the progression of the disease, really showing that you can use the organoid technology to potentially push the concept of precision oncology and improve, first of all, patient outcomes, and also potentially, quality of life for these patients. I'm going to stop there my presentation. I hope I've convinced you that the organoid technology is really the future in many different fields, including the field of oncology. And I would like to acknowledge all the people that have been working on this project, including all the Monash BDI organoid program members, people from a lab as well that are also developing these new organoid lines and the technology. The lab of Professor Helen Aber that were essential in developing some of these models, and Helen actually established the Monash BDI organoid program in 2018. Our clinical collaborators from the Department of Medical Oncology, headed by Professor Gary Richardson, our pathologists from TissuePath, and all the different platforms that support research here at Monash. And finally, I would like to thank all the funding bodies that supported our work and supported also the Monash BDI organoid program. And I'm very happy to take